afternoon, everyone. Today we're very happy to have Professor施益民from Johns Hopkins University here with TCU. Today he's going to tell about his research in cancer, okay, and 还有related to chromosomal remodeling. Okay, so Professor Shi received his medical doctoral degree from Taipei Medical University, and after that he went to University of Pennsylvania, pursued his PhD degree. And then he went to Johns Hopkins University, received clinical medical training. Okay. Let's welcome 施益民教授.
Thank you very much for your introduction, and it's my great pleasure to be at TCU again. And so, you know, I came from United States, so I just finished my quarantine requirement, and my COVID PCR test is negative. Based on yesterday, so you don't need to fear anything. Okay, so today I'm going to share with you about our recent research advancement. So I'm going to talk about the chromatin remodeling and the progression of recurring cancer. And the reason I chose this topic is manifold. One is the chromatin remodeling. I think it's a new paradigm in many biological perspectives and disciplines, especially in cancer research. Okay, so this is just introductory slides, and probably many of you have heard about and read about this book, very iconic book by Thomas Kuhn, and the book is The Structure of Scientific Revolution. And so what he tells us in the book is very simple. What is about innovation, and how can science become advanced after a couple of years. So what he proposes, this is progress in science, and probably you can explain this progress into the other fields. Okay, and this is a time, maybe decades and years, etc. So as you can see here, the progress is not a linear. Okay, it's by no means a linear relationship of your progress and the time, but instead you see this kind of pattern. So this is what Thomas Kuhn called normal science. Normal science meaning incremental advancement. Okay, you publish in this journal and this journal, and of course you accumulate the knowledge and insights. But it's not sufficient to drive any breakthrough, which we are looking for, right? So this is called so-called normal science. So this is also normal science again. So normal science, when you come to a point where you hit a wall, then you cannot further advance. And then the scientists develop so-called crisis of confidence. Okay, for example, you based on this theory for many years, and you work on that, and it seems to work because your research findings and results support of this kind of old hypothesis. But it's by no means you cannot just break through. There's no breakthrough. Okay, so you create this crisis of confidence. Then you need to think about paradigm shift. Is there any possibility that our original hypothesis is totally wrong, or we need to modify it? As a result of paradigm shift, you create a new science. So the new science, you see this exponential growth of the progress. Then after the new science, then it's autopilot again. So the progress has become normal science until the next round of the paradigm shift. Okay, so this is probably is the case for every field. So what Thomas Kuhn says is when puzzles arise that repeatedly resist solution, a crisis of confidence occurs. During a crisis, a paradigm is subject to testing and might be rejected. If that occurs, a new paradigm replaces the previous one, and the scientific evolution has occurred. So today what I'm going to tell you is maybe can be considered one of these new science, which is a chromatin remodeling. So in the past, we think, okay, DNA is a very linear. We have ATGC, we have a coding region and non-coding region, we have non-coding RNA that regulate the gene expression, DNA repair, etc. Everything is linear, but our DNA configuration rearrangement is by no means is 1D. Okay, it's not only 2D either. It's a 3D because the chromatin can be packed very efficiently and dynamically into a very small nuclei. So the DNA, if you stretch any single cell of your DNA, is about 2 meters long. It's longer than my height. Then these 2 meters of DNA, like a string, they need to fit into a very small nucleus, maybe only 5 or 8 micron. So the arrangement is very important because DNA need to replicate, DNA need to repair their errors, DNA need to be methylated, right? And also express the genes. So the chromatin need to be open and become condensed or closed all the time, depending on different functions. Okay. So back to maybe more than 10 years ago, so our team is involved in identifying ARY1A, which is a chromatin remodeling gene. And before that time, we don't know anything about chromatin remodeling and cancer. So there's 2 papers that published at the same time, and we find this ARY1A mutation occur very frequently in a lot of human cancers, especially in gynecology cancer, which is my expertise, especially in ovarian clear cell, in ovarian uterine endometrioid, ovarian endometrial carcinoma here. It's about the mutation frequency can be as high as 40 to 50%. So ARY1A become very hot research topic later on. It just become another P53 in cancer research. Okay. So chromatin remodeling, as you know, when you you took the lesson of the biochemistry, and you will know what is these structures. Anybody want to say what is these structures here? Nucleosome, right. So it composed of 8 different units of the histone molecule, right? So DNA is read by 2 and 5 times, and so the chromatin remodeling, for example, this Swiss-Nib chromatin remodeling complex, which is really huge, multi-protein complex. They have an ATPase, and they use ATP as the energy to move the nucleosome apart or move them together, or even they can eject the nucleosome histones. So in that way, it can create the histone without too many, create the chromatin without too many histones. And so this openness is important for DNA transcription, methylation, and replication, etc. So it's play a very pivotal role in basic cell biology. Okay. So this is from next year, the science paper. They do the structural biology to identify the Swiss-Nib chromatin remodeling. There are many different complexes. Okay, but Swiss-Nib is one of the largest, and the ARY1A is one of the major constituent protein. And so it's really complicated. I don't need to go over this. So you can really move the DNA into different compartmentation and configurations. Okay. So back to our research. So in ARY1A, we found out this is endometrial carcinoma. So you see, if you see red, meaning that it's a mutation. And this mutation is always deleterious mutation. Do you know what is a deleterious mutations? Meaning the mutation can lead to loss of the protein expression. And this is very typical for tumor suppressors. Okay. So as a result, and if you have a tumor with ARY1A mutation, it's just loss of this expression. And you can see there's a stromal cells or lymphocytes, they are positive because they are still have a wild type ARY1A. Okay, now we focus on this endometrial cancer, which is the major type of the ARY1A mutated cancer types, and also in uterine carcinoma is the most common female cancer. Even in Taiwan, it's not cervical cancer anymore. Maybe Professor Zhu can correct if I'm wrong. But basically, endometrial cancer, the incidence is rising very sharply because of the obesity epidemics or other nutrient factors and other microbiota, etc. But basically, uterine carcinoma has two types. I think in your basic pathology course, you must have take this kind of lessons and probably in your examination. Okay, type 1, type 2. So type 1 is so-called endometrial carcinoma. Type 2 is serous high-grade carcinoma. So ARY1A mutation only occur in the type 1 endometrial carcinoma, but extremely rarely in type 2 serous carcinoma. So I just want to focus on the endometrial carcinoma here. And you can see the endometrial carcinoma can arise from the precursor lesion we call a typical hyperplasia. Okay, then become the endometrial type 1 carcinoma. From low grade can further become high grade. Okay, so basically uterine cancer, most common presentation is abnormal uterine bleeding in postmenopausal women, but nowadays we can encounter many women before the menopause. Okay. So again, you need to remember we have a hyperplasia, that's a precursor before they become endometrial carcinoma, which is invasive. So last year we published a paper and we tried to use the whole exome sequencing to compare the hyperplasia, the precursor, and the carcinoma from the same patients. And this is what we found. We focus on whole exome sequencing to identify somatic mutations, because we believe somatic mutation can give you many important information clues, including diagnostic, therapeutic, and also pathogenesis. And this is what we found. The majority of hyperplasia and the carcinoma, they share the same mutations, meaning that they're coming from the same origins. But there are a few cases it seems to be independently developing. Okay. And we also have a case study from the same case, that this is normal endometrium, and this EC is endometrial cancer. And you see from the phylogenetic analysis, they have a different precursors, and how can this cancer development develop through this phylogenetic tree. You can see this typical hyperplasia, a type 1, and a typical hyperplasia 2, which is most close to the endometrial cancer. But AH1, a typical hyperplasia 1, is just benign, just it never become cancer. Okay. So using this kind of somatic mutation and also by computational analysis, we can be more confident to get more knowledge how this cancer develops. And so we are in the clinical team, and many of our clinical colleagues is interested in this question, whether loss of ARY1A expression, meaning the ARY1A mutation in the endometrial curettage or biopsy, is associated with the risk of endometrial cancer. And the answer is yes. So in the endometrial biopsy or curettage, in the pre-diagnostic women, and we have we perform the immunostaining, immunohistochemistry of the ARY1A, and once they are lost in tissue sections, then there is a higher chance that the patient's leftover lesion could be carcinoma, and we have a very good p-value. So in our hospital, this has become a routine test for us to biopsy. In the biopsy, if there's a hyperplasia, and if there's ARY1A loss, then I think the patient has a high risk in developing carcinoma rather than hyperplasia. So I think this can be important for clinicians to decide how to treat the patients using hormone or hysterectomy, and etc. Okay, so what's the function of ARY1A loss? And this is our old study, and you can see this one hyperplasia here, and we stand the P10. Okay, this is hyperplasia here, and this is a P10 is lost, right? You don't see any positive. AR1A is interestingly is only lost in a small portion of the lesions. And when we do the KI67, which is a proliferation biomarkers, amazingly KI67, which is a proliferation marker, is only present in those AR1A loss areas, meaning that AR1A loss can accelerate the cell division and the tumor progression. And so also we culture the epithelial cells and we knock down the AR1A. And you can see if we knock down AR1A, the cell proliferation increased. Okay, now the next question is, okay, we have the clinical sample correlated studies and also in vitro cell culture models. Then we try to use the genetic engineered mice to answer the question whether non-inactivation of AR1A, meaning deletion of AR1A in the endometrium, is associated with endometrioid tumorigenesis through transcriptional regulation. As I told you that one of the major functions of chromatin remodeling is controlling what? Transcription, right? So if there is any effect in the cancer research, we think it could be due to the AR1A loss and that remodeled the chromatin and in favor of the protein expression. So we create this mouse. It's a little bit complicated. We use the tissue-specific and also time-specific knockout of P10 and AR1A. So again, we knock out two genes. One is AR1A, one is P10. AR1A is, of course, is a new gene we focus on, but P10 is a well-known tumor suppressor, which has a deletion in very early phase of the tumor progression. So that's why we also combine with the P10. And more importantly, AR1A deletion and the P10 deletion usually co-occur hand-to-hand. So we created a PEX-A promoter-driven TTA expression. So PEX-A is the promoter controlling urinary duct epithelium, and also kidney and the thyroid glands. So if we put this PEX-A-driven RTA, okay, then RTA will bind to the doxycycline, then it will become active, then bind to the tetracycline response elements, then drive the CRE expression. This is a very standard procedure. Okay, so CRE can splice the FOXP sites. If the gene is of your interest, like AR1A, then if you're in the presence of CRE, then you will CRE out the part of gene and make it become knockout. So in that way, we will make sure that the knockout of the gene only occur in the epithelial cells of the uterus, and of course, and also in kidney and the thyroid. And we also can add the doxycycline. When we add the doxycycline, the the deletion can be complete, right? So we can decide the tissue-specific and also time-specific knockout of the particular gene of interest, in this case AR1A and P10. So the result is very simple. So when we knock out AR1A, nothing happens. There's no phenotypes. Okay, until the mice become very old, nothing happens, no pathology. But when we knock out P10, the mouse develop hyperplasia, but not cancer. But when we knock out both P10 and AR1A, and here we go, we identify beautiful endometrial carcinoma in the uterus as soon as 2 weeks after we add the doxycycline. Okay. So this is the data to show you. This is the AR1A P10 knockout. If you see red or dark purple, meaning that invasive carcinoma or carcinomatosis. AR1A knockout only, nothing happens. P10 knockout, we have a typical hyperplasia, but later on they develop cancer. But the duration is dramatically different between these two lines. And this is one example. So this is the uterus of the mouse, okay? And you see this AR1A P10 co-deletion. and as a result, they develop big uterus and is full with endometrial carcinoma. And this carcinoma spread to the peritoneal wall and the lymph node, and etc. So this is the A1A without knockout, only P10 knockout, the uterus. So the mouse uteri has 2 horns, not only one. Okay, and this is both A1A P10 knockout. You can see there's a huge uteri full with tumor, and this is only P10 knockout, A1A wild type. Okay, so we have 2 cases to show. And so this is 2 weeks after knockout, meaning we added doxycycline, and this is a uterus histology. This is a muscular layer, myometrium, and 2 weeks you can see there's a small bump. It's meaning that endometrial cancer developed. Okay. Then after 6 weeks, the tumor invades through the muscular wall and infiltrate into the surrounding fat tissue, meaning it's a high-stage disease. Only after 6 weeks, and this is another case. So this is, you see this, just have in situ lesions. But after 6 weeks, the uterus is just very, very abnormal. And under histology, they are endometrial carcinoma. So this just show you different stages of tumor progression in this mouse model. And one thing I need to mention is that we culture this A1A mouse model, and we establish the cell line. And when we put the A1A back to this A1A knockout endometrial carcinoma, you can see that reduce the proliferation and also colony formation assay, meaning A1A is a tumor suppressor. Then, as compared to human histology, as a pathologist, I can tell you that this A1A P10 deletion mouse we call iPad is induced P10 A1A deletion iPad. So you can see they are invasive. They have this undifferentiated area or squamous metapressure or angiolymphatic invasion, very similar to human endometrial cancer. But not only that, we do the transcriptome profiling, and we find that this iPad, meaning A1A P10 deleted mouse tumors, its transcriptome is very close to human endometrial cancer, but it's very different from the other human cancer types. Meaning morphologically, clinically, and gene expression-wise, this mouse model is reminiscent of human diseases. Okay, this just show you how these mice work. So if you have an extended housing these mice, they will die from their cancer. So this is a survival curve. Okay, so now we want to understand why A1A loss can contribute to tumor progression. In this case, it's endometrial carcinoma. So we perform ChIP-seq and RNA-seq in order to identify what are the genes or regions that A1A is regulating. So as a result, by combining ChIP-seq and RNA-seq, we identify several genes and their pathways may be potentially regulated by A1A. The gene and pathway including BMP4. As you know, BMP4 is very important in the TGF-beta signaling pathways. That's P4, ID4, TGF-beta1, TGF-beta2, and etc. Okay, so you can see many of the genes are involved in the TGF-beta. So of course we just focus on TGF-beta at this moment. Then we focus on DUSP4, that's P4, because it's quite interesting. It's like a molecular hub to regulate cancer gene transduction pathways. Okay. So this is a ChIP-seq showing that if you knock out A1A, it will not bind to the promoter. And I don't want to go to technical details, but basically, if we knock down A1A cannot bind to the original destined address and work as normal. So also we also try to use a knockout of BRG1. BRG1 is ATPase, but usually it is in the Swiss NIP complex with ARI1A. And so we also knock out BRG1 and ARI1A to RNA-seq and ChIP-seq. So taken together, I think this published in the last year. So if you need the information, I can send to you. But basically, this slide shows you that when the ARI1A wild type chromatin remodeling complex, they can bind to their specific site. So we identify the consensus sequence of ARI1A binding site. It's like TGA, T, probably C, and A. Okay. And so if we knock out ARI1A, so the Swiss NIP complex did not have ARI1A, what happens? It still can bind to DNA, but now it binds to different regions of chromatin. Okay, because they don't have ARI1A to find the address anymore. As a result, they still can do the chromatin remodeling, but it's quite different from ARI1A wild type Swiss NIP complex. Okay, so this is using the computational analysis, we know what's the binding sequence of ARI1A. Okay, so as I told you that many of the candidate genes are involved in TGF-beta pathways. So we try to focus on these ancient and very old, well-established pathways. And then the answer is yes. Okay. So basically, you can see this TGF-beta receptor binding promoter, and the ARI1A bind to this promoter enhancer regions. So when ARI1A is wild type, the promoter of TGF-br2, and the promoter will become more open. And as a result, the transcription machinery can work on the promoter and make more this TGF binding protein. Okay. And we also, because TGF-beta is important for many cell biology processes, including what? EMT, including tumor invasion and the migration, and etc., etc. Okay. So basically, we are not particularly interested in TGF-beta because it's well known. By this, we can connect ARI1A to TGF-beta. So that's why we published in the last year. And we show that ARI1A knockout, the cells are more motile because they have more motility, and also invasion capacity is increased using many different types of assays, including excellogens and heavy basic heating assay, or the real-time imaging cell localization analysis. Okay, so we know that TGF-beta may be important in the ARI1A pathways, right? So we applied this drug called gadolinium, meaning it's a TGF-beta pathway inhibitor. So this inacetic, we apply 75 milligrams per kilogram to the mouse, and we found that we can inhibit this ARI1A deleted induced cancer. But you may ask, then can we use this drug to treat human? Probably this is not the time yet, because this drug is still under experimental studies, and the problem this drug is very toxic. So there is no good drug yet to target TGF-beta. Although TGF-beta pathway is one of the most well-studied pathway in human cancer. Okay, so as I mentioned to you that we identified several TGF-beta related, but this one is quite interesting. It's a DUSP4. DUSP4 is a dual phosphatase. Okay, so you can dephosphorylate many phosphorylated protein. Okay, so it's US is meaning a specific phosphatase, meaning any phosphorylated protein you can remove the phosphate group. So DUSP4 is a well-known signaling transducer that can control MAPK pathway. Probably many of you have known this pathway, right? MAPK, mitogen activated protein kinase. And there are three big families. One is ERK, ERK1, ERK2. The other one is P38. And the third one is what? Yeah, right, very good, JNK, right, you are very good. Okay, so we study the command chip. So this is DSP4 region, and you can see we compare A1A wild type and knockout, and to see the chip-seq data BRG, RNA port 2, which is involving in the transcription, and also with activation mark or suppressor. This is a suppressor mark. This activation acetylation mark, and we found that this enhancer of DASP4 may be regulated by ARI1A, and actually this is a case, as you can see here, that we apply the immunohistochemistry of ARI1A and DASP4, and so this ARI1A wild type. So ARI1A wild type, you still can have DASP4 in the nucleus, right? Okay. But in ARI1A knockout uteri, the epithelial cells, you see negative for ARI1A, right? And they also lose DASP4. And this is not only one case, but we do a statistically, it's a statistically significant. And also the ARI1A, meaning and also ARI1A, the H4 of DASP4 is greatly reduced as compared to control, which is A1 type ARI1A. And the plus and minus dots, meaning we knock out the ARI1A, and this is we don't knock out. Okay, so this is another case. Okay, to show the cancer. If you have ARI1A knockout, you don't have DASP4. In contrast, in this cancer, you have ARI1A, then you have a DASP4. So I think it correlates very well. And we're very excited about this because we know DSP4 very well. And by now we know ARI1A very well. So we combine, okay, we link ARI1A chromatic remodeling and this signaling transduction mode. So I can keep telling about this is the fact, but also I did not show you is we also do the immunohistochemistry of the phosphorylated ERK1, ERK2, phosphorylated JNK, phosphorylated P38. We show the same thing. Okay, if you have ARI1A knockout, this kind of MAPK phosphorylation decreased dramatically. So we are more confident that ARI1A contribute to tumor progression in uterine cancer is partially through the TGF-beta pathway we just mentioned. The other one could be through the DASP4. Okay, so this is the hypothesis, as you can see here. Okay, so this MPK is from the receptor tyrosine kinase. After ligand binding, they transduce the signal from the KRAS, BRAF, MEK to ERK1, ERK2, which is then going to the nucleus and to perform their transcription activities. And these are PIK, P10 pathways, and so DASP4 theoretically control P38, PJAK, and also ERK1, ERK2. And so if you have DSP4 and if it's overexpressed, then you shut down this kind of the signaling transduction, right? Because remember, DASP4, the P is a phosphatase, is the function to remove the phosphate group. Okay, if we remove, then this kind of cancer signaling just stop. So that's why we think ARI1A exert tumor suppressor function through DASP4, then through the MAPK pathways. So actually, in our analysis, we all not only find that TGF-beta and DASP4 is important, but IL-6 is also very important. And do you know what's IL-6 in cancer? Anybody want to say? IL-6, very important. Inflammatory.
Cytokine.
Yeah, right. It's a very well-known inflammatory cytokines. For example, if you have COVID vaccine injection, IL-6 probably will be activated, and so induce the other collateral cytokine like IGF. like TGF-beta, TGF-alpha, IL-1, TNF, and interferon gamma and beta, etc. Okay. So this is quite important field. So AR1A is like a boss. It can control many cancer-related mechanisms. Not only TGF-beta, DASP4 by IL-6 and many others. So we are going to characterize those pathways in the future. So how about the translational research? We now know that AR1A that is frequently mutated in human cancer and how it can contribute to tumor progression. Now, here is the question: how can we use this knowledge and we can benefit the patient, right? As a physician scientist, we always think about the translational research. And so here is, we just published 2 years ago, and I don't want to go to details, but a paper showing that loss of AR1A in tumor cells renders selective vulnerability to combined ionization radiation and the PARP inhibitor therapy. Meaning that, PARP inhibitor, everybody know PARP inhibitor, right? Okay, good. Oh, we have expert here. That's great. And then because PARP inhibitor in GYN cancer may not be, is a single agent, may not be so effective. Okay, you know that. Except the BRCA1, CA2 mutated cancer. But if we combine ionization radiation, well, we find this dramatically effective, especially when the tumor has AR1A mutations. So this is like a precision medicine. We need to genotype the human cancer, whether there is AR1A deletion or mutation. If yes, we may consider PARP inhibitor plus ionization in radiation. The mechanism is shown here in the, let me see. Okay, we go to B, panel B first. If this this AR1A, go to AR1A mutant cells. So when you have a double-stranded DNA, right, you have irradiation, then you create a double-strand breaks. Double-strand break need to be repaired in order for the cell to survive and replication. So in that way, in the beginning, you create a single site break. Okay, not double-strand, single strand. So when you have single strand, you induce a PARylation and a PARP cell try to repair the single strand break. Okay, at the same time, there are other repair mechanisms involved, especially for the HR, homologous recombination, or NHEJ. So NHEJ repair is an error-prone repair. Homologous recombination is more fast-for repair. And then we have a termination EJ. Okay. So when AR1A mutant cell, then you have this, then the cell still can survive, meaning that cancer cells still can proliferate and kill the patients because they have a backup mechanism, the PARP pathways. But if we use a PARP inhibitors, then what happens is that you don't have these pathways. Okay. So like this, then you have this PARP inhibitor inhibit this PARP function. Also, PARP inhibitor can also work on to suppress or alternative NHEJ, especially when the tumor cell has AR1A mutation. Because we found in another paper, our group and others showing that the AR1A is important for the NHEJ pathways. So remember, we have NHEJ, HR, alternative NHEJ, and the PARP-related DNA repair. So if AR1A is mutated, then you don't have NHEJ these pathways. Then if you have a PARP inhibitors, also expose the cell into irradiation, then you can create a situation that you can make this cancer cell more vulnerable. So now we talk about precision medicine, right? And so the lesson we learned is not single agent will work, but it's a combination. And how you combine is not only just randomly select what are available, but you need to based on the biology, based on the theory and hypothesis, then you can better be more efficient to treat the patients in the right directions. Okay, so go back to that. Because we know that in GYN, irradiation is rarely used. in GYN, except very few like endometrial cancer or cervical cancer. But so in our team, we just submit a paper. So we try to replace the irradiation with another drug. So we screen many cytotoxic drugs, and we find one drug may be important that can replace the ionization irradiation. And this is we just submit. So next time I can update you how can we achieve the same strategy without bypassing the irradiation. Okay, so the conclusion is that AR1A emerges as a tumor suppressor. It's a new tumor suppressor, just like a P10 and a P53, etc. Especially in the endometriosis-related ovarian neoplasms and the uterine endometrioid carcinoma, meaning type 1. And we also learned that inactivation mutation of AR1A promote endometrial cancer invasion and progression in human and mouse model. And probably this is due to the transcriptional reprogramming on the epithelial cells, turn on the IL-6 and thus P4 and TGF-beta, etc. And then finally, how can we export this new knowledge for the translational studies by targeting AR1A mutation cancers with a specific regimen intervention? And so hopefully we can fulfill the spirit and the goal of the personalized medicine. Only treat those who AR1A mutated. So nowadays in oncology, there's one trend that just like a base kit therapy, right? We don't need to care about this what type of cancer, but what type of mutation or pathway they share. Then we can use the same treatment rather than they have stomach cancer, liver cancer, endometrial cancer. It doesn't matter. And we just based on the molecular culprits and prescribe the best precision medicine, regardless is the tissue type. Okay, so I just want to show you this kind of Star Trek slides. This is our view how this human endometrial cancer developed. This is normal endometrium, precursor, low-grade endometrial, high-grade endometrial carcinoma. So as we know, the P10 is occur very early. Even normal endometrium, you have a P loss, but there's nothing happening. There's no cancer, no precursor. And AR1A is here. It's not in the normal endometrium, but typical hyperplasia precursor, you have very small percentage showing AR1 mutation, but it's more and more become more important when the tumor progresses. And this is a PIK3CA similar to the AR1A pattern. And the KRAS is not so important, maybe in some high grade, yes. And PV3 is only in the high grade endometrial carcinoma, but not in low grade, not in the precursor, and not to mention in the normal endometrium. Okay, so this is our team, and our team is, actually we are recruiting postdoc fellows, so if you have any interest, please contact us, and and we can support you in every way to develop you. Okay, thank you very much for your attention. So any questions to me, and we can have a discussion.
We thanks for the Professor C's so wonderful talk. Now we open to the audience for the question. And anyone want to?
Yeah, Shi教授.
Yes.
So it sounds to me the AR1A is not specific to endometrial cancer, right?
Right. It's not.
Yeah. So it's better to be used as an early detection marker or prognostic marker?
Yeah, so this is an important question. So it depends on from which perspective you are looking at. So for early detection, I don't think this is a good marker because it's not present in the precursor lesion, only in the advanced stages. Okay. Then you want to do early detection, P10 may be better candidate. But the problem for P10 is it's already mutated in normal endometrium. So if you do that, you have a lot of false positive. So nowadays the paradigm has already shifted. It's another this kind of like stock market, okay, going up, and they will plateau. And so this kind of paradigm shift is now we cannot rely on the somatic mutation as early detection. Probably you heard a lot of blood tests, right? Circulating tumor DNA. Many people everywhere is doing that. But the new concept is that our normal body, actually we are mutated all the time. So after you listen to my lecture, your body already get about 20 or 5 new mutations in your bodies. In the endothelial cells, in your bone marrows, in your bilateral tissues. This is because every time when the normal cell divides, the cells will acquire approximately 2 mutations. So if you have more replication, then you have more mutations. Okay, go back to that. So when we are aged, we normally have more mutation, right? Even in the normal endometrium or in any tissues. So if you use a somatic mutation, it's quite terrible because you figure out your cancer test is positive. So nowadays in our team and also in many other teams, we focus on methylation. Methylation seems to be more cancer specific, also cancer type specific. And so for example, how can we, I did not bring this into the discussion today. Actually, another project we are working on is the early detection using the pap smear, the thin prep. Just like when the women visit their GY doctors, annual pap smear, right? So our pap smear is not to detect cervical cancer because the incidence is decreasing, but we want to detect endometrial cancer and even ovarian cancer. They are of the normal methylation profiles. And we also apply the microfluidic, and actually the result is quite exciting. So hopefully I can update you with this topic next time. Right.
Right, okay. Again, Yi-Ming always bring us exciting and surprise about GYN cancer research. So this chromatin remodeling seems is an important key for tumor suppressor to access to DNA, and also for DNA modification, including the repair systems. Okay, so you just presented a beautiful combination to induce DNA breaks by radiations.
Right.
And then the endometrial cells or cancer cells has to repair those damages.
Right.
So then this ARID1A Swiss-NAV modeling system play important role. Okay, so you just presented the single strand break, double strand break, basic excision repair, and NHEJ, that's a double strand break. NH, non-homologous end joining, right? So that brings to the so-called synthetic lethal. When you inhibit the PARP enzyme, then the cancer cell die. But for those normal cells, it spares because it has still one system. All right, so the same idea is that also true for mismatch repair, because you just said microcellular instability, and the endometrial cancer is one of the most vulnerable to so-called replication errors, and in microcellular, that's the marker, and also. Does this ARID1A mutation is associated with RER, replication error or mismatch repair or not? If it is a consequence, or it could be a cause, because the consequence means ARID1A may have those repeat sequences will be vulnerable to replication errors, and if it's RER defective, then it could be a target of replication error.
Right.
All right, and it also can be a cause because endometrium, as I said to students, endometrium is unique because it's keep on replicating on every menstrual cycle. So it's a tissue that create mutation error mostly equivalent to GI. So replication error or so-called DNA polymerase epsilon subunit mutations, those are very crucial for endometrial cancer development. Then, how does this relate to this ARID1A conformative remodeling?
Thank you, Dr. Zhu Tangyuan Jiao. I think this is a very typical question in journal submission. They always ask about mismatch repair and ARID1A. So as soon as we discover these ARID1A mutations and many subsequent studies showing that ARID1A mutation always go hand in hand with mismatch repair. Yeah. So there's two questions at least. One is whether this is a cause or consequence, 因果关系. You know that ARID1A is a big protein, right? 250 kilodalton, and it's really big. So if you have a bigger genes, your mutation hit will be higher, right? By chance. Okay. So maybe this is due to mismatch repair because it cannot repair the DNA. And this ARID1A is a bigger gene, right? So you can easily get hit. So that's why you have association. The other way is, no, this is because ARID1A has some regulation of mismatch repair, and it predispose the mismatch repair deficiency status like MH1, MH2, MH6, etc. And so it's always very debatable. I believe there's one more and more evidence to show it's a cause rather than it's a consequence. But in our study, we try to answer this question very early on, but we still cannot figure out. For example, we knock out ARID1A. We have a lot of ARID1A cell lines and tissue, right? And we want to study the mutation load over the time. And we find this increase the mutation load, also frame shift, but it's not statistically significant. But other people, they say, well, they find this really significant. So it depends on the cell line and the models. So I think this is still debatable, but I think this is quite important biologically. How these two different repair systems they crosstalk and merge together, and how can we exploit this kind of DNA damage repair deficiency to kill more of the normal cancer cells, but not the normal cells. So still, I don't know the answer yet.
Is there anyone who have another question?
Students and questions?
My question is about the progesterone receptors. Because a lot of type 1 endometrial cancers is treatment using progesterone. So in this mouse model, the lesions with mutated ARID1 have any relation before PR. If the combination with the ARID1A and PTEN have completely loss of the PR, or almost the percentage of PR loss in the lesions specifically.
So your question is whether the PR status, PGR status. Okay, PGR is a progesterone receptor. For those not in the reproductive science, I think ERPR is expressed in the whole GI cancer and also breast. It's very important for the physiology and also cancer development. Okay, go back to your questions. Actually, in ChIP-seq and also biocomputational analysis, we found that one of the major hub that ARID1A control is the PGR. And I'm just writing the paper now. So basically, the ARID1A loss can promote PGR loss. So this is very important questions because in many endometrial cancer treatment, right, like Zhu Jiao know the best. Now, in the early stage or in the young women, we may not take out the uterus. We may treat the progestin therapy, right, either by IUD or systemic form. And not every women will be responsive to progestin therapy. maybe 70 to 80%, but still have some significant amount of patients did not respond to progestin. So probably is this related to ARE1A because you don't have PGR, then you have progestin, then it will not work, right? That's our intuitive thinking. But actually, when we study more and more, it's not so easy because we think the progestin effect is not on the epithelial cell, but rather on the stromal cells. How the progesterone therapy works is to decidualize the surrounding stromal cells. And the stromal cell is their homeland for the cancer epithelial cells. So if the stroma is decidualized, then epithelial cell cannot survive. So it's really complicated. Not a single question can be answered directly. And so if you study more, if you figure out there's more and more questions. And so there's no question is so-called stupid, right? Even experts will not know the answer.
And the second question is about in your annual review paper, you mentioned about the prime metastasis of the endometrial progenitor stem cells during this lymphatic results. So is this ARE1A have any help for the prime metastasis from the progenitor?
Right. So the ARE1A loss can promote dissemination and invasion and metastasis is through many pathways, IL-6, TGF-beta, etc. Right? So, and you mentioned the stem cells, cancer stem cells. Well, it depends on how do you define cancer stem cells. So that field is really controversial, at least to me. And as a pathologist, how do you define cancer stem cells is really problematic. And it may be dynamic. It's not rather than you have real tangible cancer stem cell there, here, and there, you can identify. But under the conditions, microenvironments, the cancer cells can convert into stem-like cells and become EMT metastases, they differentiate again. So, and the ARE1A involved in this potential stem-like cells, we don't have any evidence, but that's the way to go, but we don't find it in this moment.
Thank you.
Thank you.
In your early slides, it showed ovarian cancer, clear cell carcinoma, has the most prevalent ARE1A mutations. But your talk today is endometrium, which is also very important. So my question is, clear cell carcinoma on the ovary is its origin is endometriosis of the ovary.
We believe.
I believe so. Then the question is, then the original source should be endometrium. And then when does ARE1A happen in this pathway? Endometrium to the ovary become endometrioma. When does an endometrioma become clear cell carcinoma?
So, great. I think this question we tried to answer about 8 years ago, and eventually we published about 7 years ago. So the upshot of our paper is we analyzed just by basic, very simple immunohistochemistry of ARE1A on a series of endometriosis or endometrioma in the ovary with clear cell here. So we compare the cyst wall of the endometriosis, not cancer, and also the clear cells, and we find that ARE1A loss in the clear cells has also occurred in the normal appearing endometriotic cyst epithelium. Not only this, PIK3CA mutations and also happen in this way. So meaning that the mutation occurred before the clear cell carcinoma is formed. Yeah, so I think some endometriotic cysts that lose ARE1A get a PIK3CA mutation may be prone to become clear cell carcinoma. Yeah, so this is the right answer.
Is there any question? Please.
Professor, it's very nice. Very happy to see you again in Tzu Chi University. My question is in the high-grade endometrial carcinoma, do you see any connection between the ARID1A mutation and the p53 mutation?
Okay, so basically in our studies sequencing, this is mutually exclusive. So basically p53 mutation is in type 2. Okay, and also in the ovarian high-grade serous carcinoma, right? Stick and this and that. But for this high-grade serous type, they have very, very rare ARID1A mutations. ARID1A mutation is one on the type 1 ovarian cancer, clear cell endometrial carcinoma, low-grade probably is not many, but basically ovarian endometrioid and ovarian clear cells. But they are very, very few has p53 mutation, about 5%. But in high-grade serous carcinoma, you have 100% p53 mutation, but very, very rare, maybe less than 1% every 1 day. So it's totally different pathways and different diseases. You can say in that way.
Thank you.
Professor, I saw that even KRAS and PAK3CA are also seen yearly. Why did you choose only the loss of PTEN along with ARID1A?
In a transgenic study.
Yeah.
Transgenic study. Okay, very good question. So we know, I can show you that this picture, right? So we know this, and you want to create a mouse model. Maybe this is a good quiz. Okay, you said, in which combination you want to do? You have a different way, right? But if my question is to understand the tumor initiation, then of course I need to choose P10 because P10 is, you see the about 70 to 80% has a mutation, right? It's a basic, 基本款, okay, basic. Then you want to PIK3CA already has been reported, I'm sorry. KRAS has been published, okay, etc. So then everyone that we discovered, and of course we want to have everyone that. Then why we want to P10? Because it's obvious, it's the most common and the most early, and that's why we choose this one. And not only this paper, we also published in JNCI many years ago. We also do a P10 knockout and ARID1A knockout in ovarian bursa, in ovarian surface epithelial cells, and we can induce endometrial carcinoma. At the same time, there's another group. They knock out ARID1A and the PIK and the knock in PIK3CA mutation. They produce clear cell carcinoma. So different mutation profile can promote different type of cancer development.
Thank you, Dr. Shin, for the presentation. My question is, as Dr. Chu explained, every month when a woman undergoes menstruation cycle, so because of rapid proliferation, the mutated cells get shed every month.
Yeah.
So why do you think that cells with ARID1A or P10 or these kind of mutations survive? Why do you think?
Survive.
Not shedding.
Not shedding.
Not shedding, yeah.
Oh, okay. Actually, we don't know whether they are not shedding or yes or no. Okay. So this is about the stem cell from the endometrium. Okay. And so we don't know where are the stem cells. When the most easy guess is in basalis, basal layer, which is not replaced every month. It's okay. So it preserves maybe the stem cell over there, right? Okay. So every month when you have menstruation, you get rid of the chromo derivative of the KRAS, or ARID1A or PIK3CA chromo, you get rid of it. Then another month, the stem cell produce, right? So the question is, if you want to make a cancer, you need to have at least 3 cancer mutations. So if you have only one single mutation, name it KRAS or PIK3CA, ARID1A, is not sufficient. The epithelial gland is totally normal, and they are not proliferating at all. It's just nothing happened to the glands because of only single one. But if you recycle it more and more, Okay, then you get trouble. Some of them, maybe 1 of 10,000, you have 2 mutations, but that's still okay. But when you get the 3, unfortunately, then you get trouble. You become hyperplasia and the tumor progression becomes cancer. So everybody have a PP3 mutation, and the whole. I think I don't have evidence. But you will not get cancer, and believe me or not, you will not get cancer unless you are so unlucky to get 2 or 3. So this is my mentor, Bert Vogelstein. Probably you heard about the name. So he called the cancer is a unlucky disease. It's just when you get cancer, because you're unlucky. That's it. It's not because you eat or you do something else. So that's theory, public science and this and that, and generate a lot of CNN report, and many people attack. But what he wants to say is biased by the media. He wants to say that if you get cancer, many type of cancer, you don't need to feel guilty. It's just by chance, just like you draw a lottery, you win the lottery. That's it. So you don't need to feel guilty. You need to pursue active consultation and treatment. And you need to live outside this kind of cancer shadow as a metaphor. And that's what he said. But actually this applied to here is beautifully illustrated. The single mutation, 2 mutation will not make any case. Yeah. And we have a lot of mutation. But what we can do is prevent this accumulation of mutations. Like menstruation is a tumor suppressor because it gets rid of 90% of the gland epithelial cell, right? And then you regenerate. You may not be so lucky or unlucky.
They also escape from immune system.
Yeah. Also, immune system play into this equation is also very, very interesting. And also, everyone that did not tell you is one of the phenomena in microenvironments increase the MDSC, myeloid-derived suppressor cells. In this, everyone that mutated cancer. It's associated with this immature neutrophil. And this immature neutrophil, MDSC, can suppress the immunity. So that's why cancer can escape and can grow bigger and bigger. So that's why we are still working on MDSC angle.
Okay, due to the time, we thanks the Professor Xu giving a special, wonderful talk. So let's thanks again. We're giving applause for the Dr. Xu.
Thank you very much.